Reabsorption is a significant part of what the kidney does. You know, in some ways, filtration is easy. Filtration is powered by the heart, right? Because the heart makes the capillary hydrostatic pressure, which pushes the solute out. So the bulk of the work of what the kidney is doing is actually reabsorption and secretion in terms of where is it spending its own ATP, right? Because filtration doesn't cost the kidney ATP. It only costs the heart ATP, right? All right, so quantitatively large and highly selective. All right, so what do we have here? We have the amount filtered, in other words, the amount leaving the Bowman's capsule, okay? The amount reabsorbed, in other words, the amount that went from Bowman's capsule back into the peritubular capillaries. The amount excreted, that's how much came out in the urine in the end, right? And then we can compare the filtered load to the amount reabsorbed as a kind of percentage, right? Okay, so we'll start off with glucose. Glucose, small molecule, freely filtered, but entirely reabsorbed in the healthy patient, right? So that in the urine we see 0. That's one of our quick and dirty tests for diabetes, right? If your blood sugar is low enough, you will have 0 glucose in the urine, which is pretty easy to test for. All right, so we get 100% of the filtered load is reabsorbed. All right, so bicarbonate, similar pattern, not quite as perfect, right? We always lose a little bicarb, but that's okay because bicarb is relatively easy to make. But there again, we see huge amounts filtered, huge amounts reabsorbed. Why? Because bicarbonate's our acid-base buffer, right? We wouldn't want to lose that because it would reduce our ability to adjust our pH. All right, sodium. We see that sodium, you know, being the primary component of osmolarity in the blood, huge amount filtered, equally huge amount reabsorbed. Now, we always have some excretion of sodium, right? But we're still 99.4%. That's still really, really good, okay? Chloride, similar pattern. Potassium, similar pattern. Now, you'll notice that potassium's filtered load reabsorption is less. That is because most potassium, remember, lives inside of cells. So the potassium load of the body is much higher than we ever see in the plasma. So we see that in a reduction in filtered load, because essentially we have to get rid of the potassium that's from inside of cells as well as from outside of cells. So we see a little bit of a less. Now, urea is a weird one. Urea is a waste product, right? In the end, what the body wants to do is get rid of urea, put urea in the urine. But it isn't quite that simple. Urea is a small molecule. It's relatively stable, and in small amounts, it's non-toxic. You actually have to have really quite a lot of urea to have there be a problem. So because of that, the kidney uses urea a little bit like it uses sodium as a way to create osmolarity. Okay, I think it's tomorrow's talk, we're going to talk about urine concentration and deletion, and dilution. The two major players molecularly there are sodium and urea. Okay, so this second to the last line will make a little more sense after we talk about that. But do know that urea, even though like it would make sense, and I know you all are becoming really good at putting the pieces together like logically. So if you put the pieces together logically about urea, you'd think it would be 0. Right? Because it's a waste product and the kidney is smart. So this is a weird one. The kidney does reabsorb some urea, even though it's a waste product, because it uses it for something else, which is to create osmolarity. Okay? And then creatinine, and remember, creatinine has like no important function in the body. All right? The only reason that we look at creatinine is it's a quick and dirty way for us to measure kidney function. Right? So as creatinine increases in the blood, that is a sign that your GFR is decreasing in the blood. So those of you who like to think in math, creatinine is directly proportional to waste products in the body, right? So as your creatinine slips up, it means your waste products are slipping up. Why? because your GFR is falling. Okay, but one of the reasons that creatinine is a good molecule for sort of tracking kidney activity is because it is filtered but not reabsorbed, right? So it is freely filtered but not reabsorbed. It's also not secreted. So it makes it kind of a weird molecule where it is only filtered. And because it's only filtered and not reabsorbed or secreted, it has a 0% of reabsorption, right? All of it enters the kidney comes out in the urine, and that is what makes it a good way to estimate GFR. We're going to talk about that at the end today. All right, so lots of reabsorption. Most of it is heavily obligate, okay? All right, so how is this reabsorption happening? Okay, we have a tubule and we have peritubular capillaries, right? So here's the lumen. On the other end of this, way up there, is Bowman's capsule, right? So this is the filtered materials coming through. And we're going to pass by the cells that make up the walls of the proximal convoluted tubule. Okay, so we have a basolateral surface and we have a luminal surface. Okay, so we think of the kidney cells as having two sides. The side that faces the lumen is the luminal surface. The side that faces the peritubular capillaries is the basolateral surface. Usually, throughout the kidney, what's on the two membranes is very different. And what's on the two membranes gives you a clue as to how this area is functioning. Okay? So here what we're looking at is we've got materials here that are being reabsorbed. What are the paths? There is a path paracellularly, just like there is in capillaries, right? Remember we talked about the intercellular clefts? Very similar idea here. Materials can move between the tubular cells, all right? And the molecules that move here are small molecules in water, so very similar to what we kind of see at the glomerular filtration membrane. So that's one path. The other path is through the cells, right? So more common or more like many of the cellular processes you've seen in your undergraduate past, right? Where we're going to have transporters and membrane-associated proteins on the luminal membrane and the basolateral membrane that are going to pull solutes through that cell using metabolism, right? So we have a variety of transcellular pathways. Active transport using ATP, passive transport. You could add into here counter transport, right, where we're using the movement of one molecule to power another, which is technically a passive transporter. And then we have osmosis, because except for the end of the nephron and part of the nephron loop, water can move in the kidney. Okay, there's a few spots where it cannot. It's actually critical that it cannot, but I'll be sure and point those out as we go. All right, so this is sort of the big picture. Paracellular path, transcellular path for molecules to get from lumen back into capillaries. Remember that this capillary came from the glomerulus, the same glomerulus that this filtration came